Cast is now starting. All attendees are in listen-only mode.
Hello, everyone. Thank you for joining our webinar, Applications and Utility of Testing Employees for COVID-19, Dissection of Myths and a Path Forward. Our speakers today are Steve Kleiboeker, Vice President of Research and Development at Eurofins Viracor, and Jaspreet Sidhu, Vice President of Technical Sales at Eurofins Microbiology. I'm Genevieve Randall, and I will be moderating today's webinar. Right, go to the next slide, please. I'm going to tell you a little bit about how this webinar will run. So the webinar is being recorded, and a copy of the slides and recording will be available within 3 business days. And the webinar will end with a live Q&A with our speakers. So to submit your questions, go into your GoToWebinar sidebar menu, select the Questions tab, type in your question and hit Enter on your keyboard. Questions that we don't get to will be answered after the webinar by our 2 speakers. And remember, a copy of the slides and the presentation recording will be available to you via email within 3 business days after today's presentation. Thank you. Okay, Steve, you can take it away.
Okay, so thank you, Genevieve, for the introduction. Just a little bit of background about this current pandemic we're in. I think most of us have maybe heard these points before, but it doesn't hurt to get on the same page. But coronaviruses, this is a large family of viruses. It's really common in people and many different animals, species of animals, that is. A lot of us have been infected with coronaviruses before the pandemic. These are the so-called seasonal coronaviruses, and they cause a mild cold, typical head cold type of symptoms, and other viruses cause this as well, but coronaviruses are one of the more common ones. But this new SARS-CoV-2, it's a different virus, obviously. It has a much more severe disease of clinical course. It is a beta coronavirus, and it has a couple of close cousins you might have heard about in the past several years that were also serious diseases that just never quite spread and didn't result in a massive global pandemic like we're seeing today. This was the MERS, or Middle Eastern Respiratory Syndrome virus, and then the original SARS, Severe Acute Respiratory Syndrome virus, back from 2003. Now, all of these viruses are thought to have emerged in bats, so that's a bit of the unifying theme here with these severe coronaviruses. This virus originated in the Hubei province of Wuhan, China. And it was thought to be initially animal to person, so a zoonotic disease, perhaps due to the open air animal and fish markets. Of course, person to person spread has been quite phenomenal since then and led to the serious pandemic we're in. And of course, it's included all countries around the world at this point. And we also know this virus has proven community spread. And it's not always known where or how the exposure occurred, and of course that greatly complicates the control measures. In terms of the disease state, the novel coronavirus, this is a new disease that hasn't been previously identified, and again, that is another complicating factor. Also, this is a virus that's different from the other coronaviruses. So any immunity that we might have built up from the common cold viruses that I mentioned in the previous slide, of course, no longer helps us out. You'll see a couple of different nomenclature names for this. The disease itself is called COVID-19, and then the virus is SARS-CoV-2, and a lot of people have just shortened that to either SARS-2 or CoV-2. This is just a schematic looking at the virus. It is a typical coronavirus. A few of the important features of this virus are the spike protein, and this is the attachment protein, how the virus gets into the cells, and of course that's how the first step of disease occurs. That's an important target for the vaccine manufacturers because if you can neutralize with antibody, for example, the spike protein, then you can prevent the virus from entering the host cell and causing disease. A few other important proteins that we look at, the N protein gene, that's a common target for many of the molecular assays, ours included. And then envelope and the hemagglutinin esterase dimer are a couple of other important targets. But overall, it's a typical coronavirus in terms of how it looks and how it's put together. So disease transmission is really important important. They're generally thought to be spread person to person through these respiratory droplets. When a person typically coughs or sneezes, and generally people have to be in pretty close contact with one another for there to be efficient spread of these respiratory droplets. The general thought is within about 6 feet. Now, these droplets can land on people's noses, mouths, et cetera, if they're nearby, and of course they're then inhaled, and that gives the virus the entry into the body. There's also a very important condition in which people with COVID-19 and are infected with SARS-CoV-2 may not show any symptoms, and these people are symptomatic. So of course they're not coughing and sneezing per se, but they still are shedding the virus, and so many people think that the efficient transmission with asymptomatics are by people touching the same surfaces soon afterwards. Well, a couple things we know about this virus, and it's been clearly demonstrated, is that it is very stable. And it seems a bit stable, more unusually stable than many other coronaviruses, so that there is certainly the possibility for people to contaminate surfaces, even if they're asymptomatic or symptomatic, and then others that come in close contact with those surfaces can also develop an infection from the transmission by that route. So that's leading to all the cleaning that you're hearing about in terms of preventing disease spread. It's also leading to the need to not shake hands and other common practices. Pre-pandemic, there were common practices anyway that could result in transmission. But really, the social distancing and the cleaning is a very important method of preventing the spread of this disease. It's also possible, as I mentioned, that for people to get COVID-19 by touching surfaces, and of course the frequent hand washing and cleaning is important there. There's also been a lot of comparisons of the current COVID-19 pandemic to the more commonly studied and more well understood influenza pandemics, and it certainly does appear that the COVID-19 or SARS-CoV-2 is at least as efficient and in many people's estimation more efficient than the spread of influenza. And so that really says something. We know from common experience how influenza can move through communities and households, and so thinking that we're dealing with a virus that's even more transmissible is fairly serious, and certainly leads or has contributed greatly to the problems we're experiencing now across the country. So signs and symptoms. I think the interesting thing, from a biology standpoint, is how varied the symptoms can be for two people who have otherwise very comparable physiology and medical history to have very different symptoms. They can be very mild, and as I mentioned before, even asymptomatic. Some people just don't have any symptoms, even though they have strong shedding of the virus from their nose or mouth. All the way up to people who are on respirators, ventilators, that is, and experience illness that tragically results in death, as we've seen for about 200,000 reported deaths in the US so far. Typically, these symptoms appear anywhere from a couple days to 14 days after exposure. Generally, they're more towards the shorter side of that, 2 to 5 days. 14 would be a fairly long time period after exposure for symptoms to emerge. The most common symptoms: fever, cough, shortness of breath, also loss of sense of taste and smell. Now this happens with other viral, especially respiratory viral infections, but it does appear to be a really common situation with SARS-CoV-2, the loss of sense of smell and taste. Certainly older people and people of all ages who have other severe chronic medical conditions are at much higher risk. Those medical conditions include high blood pressure, heart disease, diabetes are very common contributing or predisposing medical conditions. And a really important statistic to keep in mind is that the CDC is now reporting about 80% of the deaths are among adults 65 years of older, and the highest percentage of severe outcomes are even in the older group, the patients 85 years and older. And so many of you have heard from the news about the tragic situation in many of the nursing homes across the country. And so this predisposition of the older adults to the very severe, most severe outcomes with SARS-CoV-2 is really the cause of that. Mentioned that this is a global pandemic. I'm not sure if there's a country that hasn't experienced this across the world, but it certainly it's in all the major countries and regions. I mentioned also that this is a new virus, so another contributing factor to this severe pandemic is that there's no pre-existing immunity. And even if there was pre-existing immunity, it's well known that respiratory pathogens often don't elicit long-lasting or lifelong immunity. So on the opposite end of that spectrum, we can think of something like a smallpox, where vaccination, for those of us who are in the generation that receive vaccines, are typically thought to be lifelong immunity. And then at the other end of the spectrum are these respiratory viruses. So we know that the recommendation is to get a flu vaccine every year. So SARS fits more with the flu immunity scenario, in that even if there was immunity against this virus, and the vaccines that the various manufacturers are working on now may not provide long lasting, or certainly it's not likely they'll provide lifelong immunity like we think of with some of our other vaccines. These pandemics do follow a certain progression, but as we'll talk about more in a few slides, that the SARS is not reading the textbooks as effectively as we would like. So there's more severity here than what we often see with these pandemics. The peak illness often occurs at the end of the acceleration phase, and that's peak illnesses in terms of the population. And then there's this deceleration phase. We have seen a plateau or a leveling off of the acceleration phase, but we certainly haven't seen anything going back to what we consider baseline. And then also different countries, different parts of countries can experience different phases of the pandemic at any point. It can be driven by the mobility of the population, the susceptibility of the population, the weather, the social distancing habits, and just any number of factors can lead to different experiences based on the country or even the part of the country. So this is the slide that I was referring to, these pre-pandemic intervals. So this would, in terms of SARS-CoV-2, this would go back to that December 2019, January 2020 phase, where there was some initial recognition, again, and this was in the Hubei province. And then you start to see in the pandemic intervals, you start to see this acceleration where the virus really picks up a tremendous number of cases, and that's the y-axis is just a generic or hypothetical number of cases. It picks up a huge number of cases very rapidly. And unfortunately, what we haven't really seen is this deceleration phase. It looks like at this point the SARS-CoV-2 has reached a peak, at least in the US, thinking about those numbers, and now we've more or less plateaued in terms of the number of cases, and we're not seeing the downward trajectory that we like to see, or we're hoping to see throughout the US. And as we get into the flu season, where people are more naturally indoors, the cold weather months, of course, schools have reopened, both high schools, grade schools, middle schools, as well as colleges. And I think there's also some fatigue with the social distancing. We've been at this for 6 months, and you know, it's hard to keep up, it's hard to change your habits, your lifelong habits, and hold on to that. And so I think all those are contributing to a bit of a lack of a deceleration phase. And so tragically, it may take vaccines really to reach a point where the case numbers drop down to the very low levels that we see in this hypothetical graph. Polling questions here? Yeah, Genevieve?
Thank you. So we're going to pause for a quick polling question. I'm going to share that on all of your screens. And our polling question is, has your facility offered any of the following types of tests for employees? Select all that apply. Select your answer choice from one of those on this screen, or multiple as that apply. And I will leave the poll open for about 30 seconds. Okay, I'm going to close the poll now and just share the anonymous results. It looks like temperature screening has been the most popular offering for employees amongst our attendees today. Steve, you may continue your presentation.
Thank you. Okay, so hopefully that was a good overview. Many of you have heard much of that information before, but just to get us all on the same page, we're now going to switch to a new topic on SARS-CoV-2, and that involves the PCR testing or the molecular testing. Again, I think probably everybody's heard of that, but we'll dive into some of the details here and hopefully establish a little bit better understanding and a clearer picture of what we do at Eurofins in this regard. So PCR, this is an acronym that stands for polymerase chain reaction. There's a slight modification to the standard polymerase chain reaction.
I'm going to pause you really quick. It looks like the poll is still showing on your screen. I'm just going to close that out. And just a minute. I'm actually going to share my screen instead, and then you can continue your presentation.
Okay, so you have this.
Yes, sorry for the slight hold up. Okay, and now you may continue. Just let me know you'd like to progress.
Okay, yeah, so you can advance one slide. Yeah, so as I said, PCR, actually that's the basic method. This looks at DNA. Obviously, this is an RNA virus, so we have a preceding step to PCR known as reverse transcription. I think the most important point to think about with PCR is this is one of the most sensitive analytical methods when looking at RNA and DNA. It does provide the ability to detect just a few copies of the RNA and DNA. And then, of course, it produces through amplification, the chain reaction that is, billions of copies. And so you can easily detect the signature, if you will, of the virus in a very short amount of time, really just a few hours. So this is, as I said, one of the more sensitive methods, actually the most sensitive method for SARS-CoV-2. It's not perfect, however. There are a few limitations. One is inhibition, and that is just a phenomena that samples. We do include controls with each and every reaction, so that if there is inhibition, we know that, and we avoid reporting a inaccurate or false result. Also, one of the important things about PCR is it does require sequence information to design an assay. These are the primers that you see on the screen there. We live in a really remarkable time in that the sequence of SARS-CoV-2 was available within just a few weeks of the virus being identified, and it was shared throughout the world. And so at Eurofins, at that point, we didn't know it was going to be a pandemic, of course. That was back in the early January timeframe, but we thought it was certainly possible. So that having that sequence information allowed us to get started developing an assay, hoping we'd never need to use it, but always being prepared and at least a step ahead of a virus or a pandemic like this does have a significant advantage. Next slide, please. So we'll talk a little bit about the testing broadly. So this, as you've seen, I'm sure, in the news many times, this SARS-CoV-2 testing is available through the public health, the Centers for Disease Control, many state public health laboratories, and then also a really large number of private laboratories. So across the U.S., this conglomeration of various laboratories has come together to provide literally the capacity to do hundreds of thousands of tests. I'm not sure what the total number of tests that have been run yet, but it's well into the tens or maybe even hundreds of millions, and it's just all coming out of these laboratories doing 10,000 a week or 5,000 a week, or some are doing as much as 100,000 a week in the really high throughput lab. I'll talk a little bit about the assay that we've developed at Eurofins, specifically at the Viracor Eurofins lab. And then one important thing to remember is that this test is qualitative. Now, A quantitative test would have some advantages, but at this point, these tests are generally regulated by the FDA through what they call an emergency use authorization process. And you're probably familiar with that through the drugs and the various treatments that have come out. Those are regulated under the same process. So we are capable of reporting only qualitative results now. So that's a distinction that I think we will see evolve over the next months in terms of dealing with the pandemic. And there are also a number of different specimen types. We've talked a lot about nasopharyngeal swabs in the community, but nasal swabs and saliva are becoming more prominent for testing. These certainly have the advantages, the nasal swabs and saliva, that is, have the advantages over the nasopharyngeal swab in that they're less invasive and not painful to collect, and generally provide very accurate results. But this is all listed on the Eurofins Viracor website in terms of the details. Next slide, please. So interpreting the results, there are really two results that come out of molecular testing. And so the positive is referred to as detected. And what this indicates is that at the time the sample is taken, SARS-CoV-2 is present in the sample. And so that means that the patient, the individual, if they're asymptomatic, is actively shedding the virus, and that the test that was run, the RT-PCR test that was run, did detect presence of the viral RNA and therefore the presence of the virus. Conversely, not detected, that means that no virus was detected in the sample, no SARS-CoV-2 was detected in the sample. Now, this is generally a very accurate result, but it's important to point out that the virus is not shed consistently in some patients. They'll have periods where they're not shedding before they start shedding, and so that's the so-called eclipse phase between infection and the shedding of the virus. They can go on to shed the virus a day or 2 days or even a few 3 or 4 days later. So we have to think about the biology of the virus when we interpret these results. And then also patients may be over an infection seemingly, and then they may start shedding again. And I think that there have been several reported cases of patients who actually got reinfected. So they had the infection, they may have tested negative for a while, and then they get infected a second time with the virus, and so then they could be positive again. So there's always an important need to think about the patient in general, think about the individual in general, if they were exposed, before you just blindly interpret results as either positive or negative or detected or not detected. The CDC's done a really good job of putting together guidelines, and so the medical doctors, medical professionals are following those. Of course, our laboratory follows those in terms of the interpretation of these tests, and it has been quite a useful resource. There is also the need to report these results. So all laboratories testing, including Eurofins labs, do report the results to the local and state health departments and public health officials. Next slide, please.
Okay, we're going to pause for another polling question. So I'm going to launch that on your screen now. And that question is, have you been tested for COVID-19? Select your answer here, and I'll just share anonymous results on our screen, and then we'll continue with the presentation. Okay, I'm going to go ahead and close the poll now and share the results. Looks like most of you have not been tested for COVID-19, and so we are going to go into a follow-up polling question, and that is, if you haven't been tested for COVID-19, why? And select all that apply. And your polling question should be on your screen now. I'll leave the poll open for about 30 seconds, and then we'll continue with the presentation. I'm going to go ahead and close the poll now and share the results with you all. And we're going to go on and continue the webinar.
Okay, thanks. Next slide, please. Okay, so the next section we're going to talk about is antibody testing. And there's some really important distinctions between the antibody testing and the RT-PCR testing that we just discussed. I'll try to make those clear as we go along. Next slide, please. So just as an overview, and I'm going to lump in the antigen testing along with the antibody testing because there are some parallels, especially in terms of the ability to interpret the results with some level of definitiveness. So antigen testing is actually somewhat similar to the RT-PCR testing in that you're testing for the presence of viral proteins. Now remember, we talked about RT-PCR testing viral RNA. The antigen testing looks for viral proteins. Similar thought there. If you have viral proteins, that's good evidence of a current viral infection, just as if you have viral RNA, that's good evidence of a current viral infection. The huge difference between antigen and PCR or RT-PCR testing is the level of sensitivity. And antigen sensitivity is many thousand times lower or worse than RT-PCR testing. So the antigen testing can be a good screen, and there certainly is the ability to detect very high levels of virus being shed by an individual, but the challenge with antigen testing is that a negative is very difficult to interpret. You don't know if the patient doesn't have virus or if it's just below the limit of detection or the level of sensitivity really of the test. So the use of antigen testing is limited by that factor. If used properly, it can be a nice adjunct to a testing program, but there are more pitfalls than we see with the RT-PCR testing by quite a large margin. Now the second category, the third column here on the page, looks at antibody testing. And so antibodies are part of the immunological response that a person mounts against the foreign invader. In this case, of course, we're talking about SARS-CoV-2. So SARS-CoV-2 antibodies in a patient indicates that the virus has been present and that the individual has mounted an immune response, and the antibodies are part of that immune response that we can then detect with the various assays that are available. Next slide, please. So there are actually two types of antibodies that you'll hear discussed, IgM and IgG. IgM is the early antibody. It's produced typically within a few days of when a person gets infected, and then it also goes down relatively quickly, sometimes as early as 10 or even 14 days post-infection. IgG is a bit later, although paradoxically with SARS-CoV-2, many times IgG and IgM come up together and they go down together. And that is part of what makes the interpretation of antibody tests challenging. So the IgG is a smaller molecule. The size really doesn't matter. Obviously, you can't see any of them, but the key about IgG is it should give you a longer term response. And in some cases it does. It's just not as consistent as we typically think of when we think of measuring the immune response to viruses and other pathogens. So next slide, please. So again, IgM, this is expressed on B cells. These are the cells that are going to make the IgG. IgG has several forms. I think this is probably not as important in the interpretation. I think the key to remembering the difference is IgM is early and IgG is later, or at least it should be. IgG can cross the placenta, so I've not read about this, but certainly if the mother is infected during the gestation, there is the potential for the baby to be born with some passive immunity, some IgG to SARS-CoV-2, but again, I'll emphasize this a couple times. The IgG levels typically have not stayed elevated for long periods of time, even in patients severely infected with SARS-CoV-2 and who recover and go on to be healthy and normal. The IgG levels tend to drop off fairly quickly. Next slide, please. So this looks at a stylized curve or response curve in terms of the timing for IgM and IgG. If you look at the very first part of the graph, the antigen, that's the protein that induces the IgM and IgG. That's the SARS-CoV-2. And as I said, typically IgM comes up quickly, goes down relatively quickly, and then it's followed by IgG. But what we've seen in many of the studies that have come out with SARS-CoV-2 is that sometimes IgM and IgG come up closer in time than we typically see, and then they also tend to both go down quicker than we typically see with other pathogens. So what you see on the screen here is the patients who are doing the right thing with their immunological response. Of course, we don't have any control over that, and it's not broadly applicable. We see a lot of variability with SARS-CoV-2 in terms of antibody responses. And at the very heart of it, that's what makes the interpretation of immunology or immunological responses, antibody responses to SARS-CoV-2 difficult, is that there's just a lot of variability. And then additionally, we don't always know what protective levels are, especially at the early stage. So there's a difficulty even in interpreting this. If you have a patient, for example, that is had classic symptoms of SARS-CoV-2, the patient may have tested RT-PCR negative. The sample may have been collected late, for example. And if there's still good reason to think that this individual was exposed to SARS-CoV-2, and it's important from an epidemiological or a medical reason to work further to achieve that diagnosis, then an IgM and IgG antibody test can be very useful in that scenario. Another scenario where they're useful is just looking at a population as a whole. What is the exposure history? And so while the assays are not always accurate within every individual in terms of telling us about the history, certainly if you test enough people in a population, say 10, 15, 20 or more people, you're going to get a more accurate picture, because some of those individuals will have had a more typical immune response that can be interpreted cleanly. Next slide, please. So a little bit about test methodologies for antibody testing. There are quite a few out there. You'll see the various acronyms here for the enzyme immunoassay, radioimmunoassay, et cetera. As with, or unlike, I should say, the RT-PCR, where there's really just one common method, these other methods are really just different from a laboratory standpoint. They still seek to measure the same process, and that's the formation or the production of antibodies by the body. A couple different Eurofins labs are running two of these methods. The Boston Heart lab uses a chemiluminescent immunoassay, and then Viracor uses the enzyme linked immunoassay, or the EIA. Next slide, please. So this just shows you a little about interpretation. Now, we didn't talk about values with the RT-PCR, although I mentioned that they will be useful, and I think they will play a role long term, especially if we're still dealing with this pandemic in the coming months and years, which it does seem likely at this point. But conversely, with antibody testing, we do provide a value, and so this just looks at an example of an interpretation of the assay that Eurofins Viracor runs. A value of less than 9 indicates that there's no detectable antibody present. Now, remember, this could be for two possible reasons. Either the individual is not exposed, or it could just be early. Think about that time graph, the stylized time graph that I showed a slide or two ago. And the period of time between when an individual is exposed to SARS-CoV-2 and when they actually mount a detectable antibody response is somewhat variable. It can vary anywhere from 5 days to 15 days. And remember, we don't always know when an individual was exposed. Sometimes you may have a good history and say, okay, well, this person was tested positive, was symptomatic, and this person came in contact with that individual. But many, many cases occur with no real definitive exposure. So even if you know the time, of course, the time to mount an antibody response is variable. Values for the EIA tests at Eurofins Viracor of 9 to 11, that's a gray zone, an indeterminate zone. There is potential evidence for antibody infection. In many cases, it's just early on. So you're just seeing that early rise. You don't have a real strong signal. But if you test that individual, say a week later, then you would have a strong and definitive positive. And then values greater than 11, there's pretty clear evidence that the virus was there. Again, the diagnostic accuracies here are going to be in the high 90s at this point. But of course, if the patient is tested many weeks after infection, then that antibody response could actually have already come and gone. So you may have quote unquote missed it in terms of the ability to get that greater than 11 in this example. So what to do when an employee tests positive? So if you're looking strictly at an antibody test and you're wondering about a patient's ability to transmit, you can't determine that from the antibody test alone. That would typically lead to some follow-up testing with RT-PCR, because if the person has been exposed, there is a potential that they are still shedding the virus. In some remarkable cases, people have shed the virus for 6 or 8 weeks, and it's really unknown how that occurs. It is unfortunately not common, but it is possible. And so a positive antibody test may lead to additional PCR testing. Again, there are really good guidelines that the Centers for Disease Control put out, and the results also do need to be shared with the state and local public health authorities. Next slide, please. So just to reiterate, antibody testing identifies people who were exposed and developed a potential immunity. You'll see the word potential italicized there. We don't always know in many disease states, and especially with SARS or COVID-19, we don't know what an immune level would be. Recall I mentioned a few slides back about people becoming reinfected. Did they have no antibodies, or did they just have very low levels? There really haven't been good studies to point that out yet. So oftentimes the antibody levels are important for healthcare workers because they are being continually exposed. But of course, identifying potential immunity could decrease the overall number of people in quarantine and help them get back to work sooner. There haven't been clear cutoffs and guidelines set though, so that's something that has to be really worked through with a medical professional on a case-by-case basis, and the programs are the best programs that are looking at this are using a risk-based sort of assessment. Antibody testing does give a really good long-term picture of the patients, especially when it's used on a population basis, or not just patients, but workers as well, especially when it's used on a population basis. And then there are some early promising results where the antibodies produced by some patients, those that have been infected and recover, can actually be used to treat other patients. And so that's actually in use in certain facilities that does require the emergency use authorization that we talked before previously about therapeutics and the various testing methods. But don't confuse that with the screening of patients. The therapeutic use is typically different or different consideration than testing populations for their exposure to get an idea, I should say, of their exposure history. Next slide, please. Okay, so the next section, we'll talk just a little bit about pooled testing. And so this takes advantage of the really high levels of sensitivity of RT-PCR and can provide a more efficient and cost-effective method of testing large groups of people simultaneously. Next slide, please. So the idea is pretty simple. With pooled testing, you're taking multiple samples and you're combining them into one single reaction. And of course, that both extends the capacity of labs. And many of you have heard, I'm sure, about the fact that our labs are stretched thin at this point. There's very little excess capacity. Many labs are straining under the load, the massive number of samples that require testing. So it extends capacity by combining a number of reactions into a single tube. And of course, that also has the impact of reducing cost. Next slide, please. So as with all things SARS-CoV-2, the EUA process is required. So at Eurofins Viracor, we have submitted a request for approval to the EUA, to the FDA through the EUA process. We have gone through their thorough review process, provided some additional responses as needed, and we're expecting that to be completed very soon. I mentioned the combining of the samples. One important point to remember is that The method does need to be very sensitive. We include the same internal controls, obviously, that I think I mentioned previously as well. But by having a very sensitive method, we can combine up to 5 samples and not lose the ability to detect a single positive. Next slide, please. So this just looks graphically about how this would work. So we show the 5 people here all collected with a single sample. The samples are then combined at Viracor's lab, and the RT-PCR method is performed on that combined sample. Now, if the pooled result comes out positive, we do typically determine which of those 5 individuals, or potentially more than one of those individuals, contributed a positive sample. So we retain the original samples, the original separate samples, and then if the pool is positive, we go back and test those individual samples to see which of the individuals are positive. However, if the pooled result, that is, comes back negative, then it can be safely assumed that all 5 individuals who contributed samples to the pool were also negative. So that's a nice clean result that doesn't require any reflexing. Next slide, please. So as I said, if a pool result is positive, we retest to determine the individuals. These do, in our test code language, these do have separate numbers so that we know the process that's being requested. Turnaround time is really important. We haven't talked about that with the PCR testing, but I can't emphasize enough the importance we place in our lab on returning these results typically within 24 hours. Now, if there is a pooling, there will be some additional work required, obviously, for positive samples, so that can add a few days. But just overall, a rapid turnaround time is key. I'm just thinking about the usefulness of the information. The value of information about who's positive, who's not, declines dramatically after a couple of days, primarily because some of these individuals will have been out and contacting other individuals unknowingly the whole time that they were positive. So I really would emphasize the need for all of us to focus on rapid turnaround time to really maximize the value of RT-PCR testing. Next slide, please. So I mentioned the cost effectiveness of this as also the efficiency of utilizing the strained laboratory resources in many cases. And then also the sensitivity of the assay does provide the same quality of result, essentially. We saw a slight degradation in the actual machine readout, but by adjusting our cutoff, we were able to maintain the same high quality and sensitivity. And pooling is just one of the many aspects in Eurofins clinical diagnostics efforts, along with antibodies, surface testing, wastewater testing, used mask testing. All of these help build a program that can be used to understand the spread of the virus, the risk of spreading the virus, and obviously significantly reduce the impact. The pooling is most effective in groups with low prevalence. If you look at the math, if you will, you have to have a prevalence of below 3 to 5% in a population to really realize the efficiency of pooling. If it's too high, then you just end up breaking out a bunch of positive pools, and you can actually end up doing more testing, not less, if you have the prevalence of, say, above 5%. And then since it is cost effective, it does provide an opportunity to do extensive surveillance of individuals during the quarantine period. And many times the quarantine, we're looking at 14 days, but you do need typically a negative test to effectively exit quarantine. And so this can provide that sort of a strategy on a cost effective basis. Next slide, please. So this just looks at the overall safer at work solution. It really is an overall long-term effort that provides comprehensive testing in terms of strategies and practical approaches. Involves consulting services. We've talked a lot about the PCR testing as well as the antibody testing. Haven't talked as much about the surface or environmental and more mass testing, but those are all part of the picture that we see that allows employers to effectively assess and keep their employees safe in their place of work. Next slide, please.
All right, so that is the end of the presentation portion. We're going to do a quick polling question, and then we'll go into a live Q&A so that Jaspreet and Steve may answer your questions. So I'm going to launch the poll now. And that question is, do you want a representative from Eurofins to reach out to you regarding services discussed in this webinar? I will keep the poll open for about 30 seconds and we'll move into Q&A. Okay, I'm going to close the poll now and we'll move into Q&A. And our first question is, why would you test employees if you haven't had an outbreak?
Yeah, I'll take a shot at that. That's a really good question. So testing is most effective when there is some reason to suspect some sort of exposure. It can be in your community, it can be within individuals themselves. Quite honestly, there aren't too many communities that haven't been exposed at this point in the US. There probably are some pockets where the prevalence is very low. But I would emphasize that in terms of controlling the impact of SARS-CoV-2 on groups of people, the sooner and earlier, I should say, the earlier really is the key. The earlier you can identify infected individuals and just keep them separated from the rest of the community, the more effective the control measures will be. When you have a 20% positivity rate, for example, none of the control measures work very well. At that point, you're just waiting. But if you can identify individuals, especially if they have higher risk settings early in the process, then that will decrease the impact and shorten the period of control efforts in a workplace, in a healthcare facility, in a school, any of these places where groups of people are congregating.
And I can actually add to that two other examples that are really relevant in today's culture, one being the NFL and how rapidly they've had to respond when two teams have one individual here and there has tested positive. It leads to an immediate shutdown of their operations. The White House and associated staffers, that's another very good example where rapid testing is being enforced on a daily basis just to stay ahead of any potential outbreak. So really, it's not just a matter of an outbreak, it's just remaining vigilant and ensuring that when something does occur, you can take an immediate action.
Thank you for your responses. Okay, our next question is, what can employees do outside of work to prevent outbreaks in my facility?
I can address that, Steve, if it's okay.
Sure.
From a microbiological perspective, the practices that are implemented inside a plant, say a food processing plant, are really valuable in teaching employees how to administer good sanitary practices while processing food, while producing food or handling ingredients, raw ingredients. Same goes for a restaurant or for a food service environment. The practices that you are teaching your employees and your employees are exercising at work are very easy to carry over into a home or a private environment, such that effective hand washing, not touching contaminated surfaces, or not contaminating surfaces when they're touching their mouth or their nose, and sharing utensils, etc. So it's really common sense is the best way to describe it, but please know that the GMP and SSOPs that you're following or expecting your employees to follow at work should easily translate over to their practices at home. And it's a circuitous effort to make sure that they stay healthy while at home and also stay healthy in the work environment.
Yeah, I second all that. I would emphasize also just the vigilance. I'm a virologist by training and obviously work in the lab industry. But for me at this point in time, I believe the best attitude is if you have upper respiratory symptoms, it's SARS-CoV-2 until proven otherwise. If you have a fever, it's SARS-CoV-2 until proven otherwise. I think if as a community, as a country, as a as an employee group, if we all took that attitude. Now, how many of us over the years have gone to work when we have upper respiratory symptoms because we don't want to miss that meeting, we don't want to get behind, we have important stuff to do. So that's the past. I did that. I've done that before. I think it would be irresponsible to do that in this current environment. So just personal vigilance for your kids, for your dependents, for your those who you can influence around you, can go a long way to helping out this pandemic in a very practical sense.
Thank you. Our next question is, are we concerned about pooling samples that will lead to confusion and miscommunication?
So that really is on the lab. And we at Viracor Eurofins, we have extensive processes in place to prevent this confusion. I would emphasize that the modern labs have what we call positive identification throughout, through barcodes of plates, of racks, of tubes, and the pooling doesn't add significant complexity to that. Some, but not significant. And with positive ID throughout, once we create a pool, we know the members of that pool has a positive ID throughout every step of the process. It's not an unlabeled tube and an unlabeled rack being carried across the lab. There are very strict measures in place. So while nothing is perfect and anything bad can happen, I'm very confident that the measures that we have in place will prevent these sort of confusing events that the question alluded to.
And I'd like to add to that, Steve, in the traceability of our analysis, pretty much every sample is barcoded or receives a barcode. So there is full traceability throughout the entire process. And in the food industry, a really good analogy is when you pool samples or you make composites, and then if a positive occurs in that pooled and or composite sample, it's very easy, or should be very easy, for the lab to go back to which of those samples it originated from. And having barcoding throughout the entire process makes that process a lot more infallible. And certainly we reduce the likelihood of cross-contamination and or mixing of samples.
Thank you. Okay, our next question is, what is the typical number of employees needed for an effective group test?
Yeah, I don't know that there is a low number or a high number. I think it really is a risk-based assessment in terms of potential exposure, the environment that you work in, the community that your currently employees are living in. Even a few employees, if they are working in close contact with one another, of course, one person can spread it to 5 people quite quickly. And then, of course, I think common sense tells me that the larger groups probably have more room for damage, if you will, in terms of, say, infecting 20% of 500 people is 100 people, as opposed to a very small number. But still, nonetheless, in a good program, a consultant can look at the risk of these individuals and come up with the most common sense and effective way of screening.
I would agree with that, Steve, and also add in a process environment or production environment where there are multiple shifts and constantly changing shifts, it's critical to note that one person may carry over, and so a second shift would essentially undergo the same level of contamination risk if that same individual was posing a risk to the first shift. So risk assessment is really simply the name of the game here, and it centers around just how complex or how people-centric your process is, and the likelihood of exposure increases with the more individuals there are on site at any given time.
Thank you. Okay, our next question is, how is Eurofins test different from other tests on the market?
Yeah, so the RT-PCR test that we've developed is one of the most sensitive tests available. We think that's important for a couple of reasons. First of all, in a pandemic, in a situation where the repercussions are so high, having a false negative due to poor test sensitivity is really very challenging in terms of controlling the infections in the pandemic. So a highly sensitive test has ideally no false negatives. There probably is no perfect test, so we've made our test as sensitive as we can to reduce the false negative potential. And then also having a highly sensitive test allows us to do things like pooling without a loss in diagnostic sensitivity. So it's really a matter of, again, accuracy and then the options that you have for designing group tests such as a pooling strategy.
And if I may add to that, in addition, our test is considered the gold standard in regard to transplant tissue acceptance, since that's where a lot of our adventitious agent testing was established in maintaining the integrity of transplant tissue prior to
Jaspreet, we might have lost your audio. Okay, we're going to continue on with the next question, and Jaspreet, if you're able to join us, you can come back in and finish your response. Thank you. Our next question is, can all of the tests in this webinar detect asymptomatic positives?
Yeah, the simple answer is yes. The biology, obviously, of disease is different between symptomatic and asymptomatic individuals, but fortunately, the RT-PCR test and the antibody tests work similarly on the two various groups. And of course, those in the middle, they're the mildly symptomatic as well. So easy answer on that is yes.
All right, excellent. So that is all of the questions that we've received today. Thank you so much to both of our panelists that were on the webinar. And I want to remind the audience that you will receive an email within 3 business days that has a copy of the presentation along with the recording from today's webinar. If you would like Eurofins to reach out to you, you may contact us at our website, which is on this screen. Thank you for joining and have a great rest of your day.
Thanks, Genevieve.